Good morning, everyone, and welcome to our 11 o'clock briefing. Long-duration spaceflight raises human health concerns. We have 3 speakers on our briefing today. They are Craig Kundrat, Director of the Space, Life, and Physical Sciences Research and Applications Division at NASA headquarters, Christopher Mason, Associate Professor of Computational Genomics at Weill Cornell Medical College, and Sylvan Cost, GeneLab Project Manager at NASA Ames Research Center. Without further ado, we'll get started.
All right, thank you. The purpose of my talk today is really to provide context and set the stage. So I'll make my remarks here brief. There's to give you a heads up, one bit of embargoed information coming out later today that I'll tell you about in a moment. So the context is the exploration campaign, which you see on the slide behind me here. NASA's plans are to continue to use LEO for the foreseeable future that's low Earth orbit. I have to remember to speak English, not NASA. So to use low Earth orbit for research and technology development, but we're pushing ahead to go back to the moon and on to Mars. And around the vicinity of the moon, the 2 major pushes are developing a orbital gateway and a series of lunar landers. And a lot of this work is being done in preparation for human missions to Mars, to learn what we need to do there, because the moon is only a 3-day trip from Earth and vice versa. So if things don't go so well, it's easy to get back. Whereas Mars, it's a 6-month journey, and when you work out the orbital mechanics, there is no fast return. So we really want to use the moon as much as we can to learn the things that we need to go on to Mars. I'll describe the 5 major areas of stress or hazards to human space travel. That's altered gravity, radiation, closed environment, isolation confinement, and distance. And I'll go into those more detail in the talk, and I'm happy to answer those in questions. That'll set the stage for Chris, who will be diving deep into part of our human research programs, one of the projects in that, the twin study, which is looking at the human response to spaceflight. And then Sylvan will follow that with a talk on GeneLab, which is part of the space biology program at NASA. And that space biology program looks at a wide range of organisms, like microbes, plants, as well as animals, and typically looks much deeper at mechanism than the human research program is able to. And GeneLab is a very powerful capability for that. And Sylvan will describe that. The bit of embargoed information coming out later today is the space biology program is announcing the selection of 4 research investigations for, in NASA speak, EM1. That's Exploration Mission 1, which is slated for launch in 2020. That will be about a 26-day mission. So this will be the Space Launch System rocket that NASA is building, along with the Orion capsule. It will be uncrewed, but it'll have payloads aboard, including these 4. And it'll be about a 26-day mission, so it'll transit out to the moon, do a series of orbital maneuvers, and then return. So you can look for that press release later this afternoon that will announce those 4 selections. And the purpose behind these investigations is really to look at the combined effects of microgravity, because that would be the gravity environment during most of the mission, and most importantly, deep space radiation, which is quite different than the radiation we encounter here on Earth. Pretty much everything in terms of ionizing radiation we encounter on Earth is X-rays or gamma rays, which are a form of light. And in space, you are exposed to atomic nuclei, from the lowly hydrogen up to the heavy iron. And so the biological damage is quite different, and that's what the object of study is in those coming up that you'll learn about more this afternoon. And with that, I'll turn it over to Chris.
Thanks. Well, thank you, Dr. Kundrat, and it's a pleasure to be here today. Thanks for everyone for coming, and you'll hear, of course, as Craig just detailed, there'll be some brief remarks now, but also a longer talk I'm giving later today. And let's see if this, the first thing, the other one, I guess the other slides we've got. I just had a couple images just to display, which is a little bit about the study, and really I'm one of the investigators. There were 10 teams chosen, you can see here, as well as many collaborating labs to help out with the NASA twin study, which was really started in 2014, and so is this idea, well, It didn't start as a blue line, but that was only when things went wrong. But it'll come back up. It'll come back up. The goal was to really leverage a very unique opportunity. So to understand what happens to the body during a long duration space flight. In this case, a year in space that Scott Kelly was about to launch and did in 2015. And as a geneticist, I was very excited because my training's in genetics. All I do all day is think about genetics and DNA and RNA and what's happening inside cells. And so I wish every experiment that was ever done had an identical twin with it. And so this includes anytime you send someone to space. So we were very fortunate that Mark and Scott, Mark, who is actually retired and still is, although now is running for Senate. So that an interesting development in the last week. But he was retired at the time, but volunteered also to help with the study, to have blood collected, and gave literally blood, sweat, and tears, or some tears. He probably doesn't cry that much, I'm not sure, but gave a lot of fluids to the study, as well as Scott, to bring one to space. So really, everything that we could ask for, they were both really great, basically study subjects. And so I'll be presenting some of the results of that study today. I'll give you some brief highlights now, and then I'm happy to take questions, and also I'll be in much more detail this afternoon for about a 25-minute talk. But in a nutshell, we looked at everything we possibly could. And again, not just me, but this was a collaboration, really a group effort that's actually also spearheaded by Craig and John Charles and historically Graham Scott, was to look at these two identical twins and measure everything that we possibly could. Because in the face of lack of knowledge of something, when you don't know what will change, the best thing to do is to measure everything that can change. So we measured, I'll describe a little bit today, most of the work from our lab on the genetic and some of the epigenetics and transcriptional changes, or the gene expression changes that occurred during spaceflight. And some of the embargoed information that's coming out today details other things we've seen about the mitochondrial fraction of DNA that showed up in Scott's bloodstream when he was in space. So this is also something you'll hear about in GeneLab, is that we see evidence of mitochondrial changes in spaceflight that we didn't expect. Other things that have already become public, you've heard about probably, are that telomeres got longer in space, which we also didn't expect. We thought maybe the opposite would happen. So several surprising features of the study some have come out, some will come out today. Another thing is we looked at the distribution of cell types, so what numbers of cells are appearing in the blood, particularly in the immune system, where we see the immune system changing very dramatically during spaceflight. So that it appears as if, almost the body is on kind of a high alert, which you would imagine if suddenly you were in zero gravity, your body would probably be on high alert as well. But as far as we know, all these changes are really adaptations and actually really response to spaceflight. Actually, one of the bits of really good news for today is that it's mostly really good news, actually. Like, of all the changes we see, a lot of this is in manuscripts that are in preparation now and some in review, is that we see a lot of things change in space. Thousands and thousands of genes change expression, molecules change in the blood, what are called metabolites. We see changes in the microbiome. We see changes all across human physiology, but over 91% of all the genes return back to normal very quickly upon coming back to Earth. And we see that for the most part, the body has this extraordinary plasticity and really adaptation to being in zero gravity, at least for a year. That's what I'll present a little bit today. And then also we'll talk about plans for future missions. There's additional missions planned that will be long duration missions over the next 7 years. And so, of course, a sample size of 2 limits what we can say, where this is not the be-it, end-all definitive study of human space biology, but it is really is the most in-depth molecular portrait, I think, of almost any human being that's been created in a lab or at NASA or really anywhere. So it gives us really this extraordinary first contours of sort of the landscape of what changes in the human body. And as we describe in some of our writings on this work, is that there's things that are very plastic that we don't seem to need to worry about too much yet because they change, but they just seem like adaptations. Some things that seem to persist maybe out to 6 months, which I'll describe this afternoon, in gene expression. They're kind of just things to keep an eye on. They're not good, they're not bad. We just know that some things changed, and we'd want to monitor those things that change the most, and then replicate this in other studies and animal models. And so I'll present that this afternoon. I think the last slide is just, I think that was it. That's Mark and Scott. And I want to thank them as well for being a great subject for the study. And this is the teams. This is 10 different investigator teams that all collaborated very closely over the last 5 years. And we're now just at the very end where we finished all the experiments, finished most of the analysis, and other writing is done for the first paper, and there's several papers in draft form. So it's you should see a lot of things come out this year and some coming out today. And thank you very much.
Okay, so I'm going to follow up with as we heard, we have a video. I'm going to, maybe we can show it in 1 minute, but just to give you a little bit of an introduction of what my project is about. GeneLab is a database that basically is collecting all the different omics, all the samples we have in space, and also samples done on Earth can be processed through omics technology. And so this is a revolution in biology right now. You can do genetic, you can do transcriptomic, you can do proteomics. And so we're really looking at molecular changes in the tissue. So as we heard, you know, a lot of fluid were taken from the astronauts, but that's not enough because those are non-invasive methods. So you can only grasp some changes when you look at blood, when you look at non-invasive method. When you start working with animals, you can now really go into very deep in the tissue, and basically you can collect whatever you want, heart, muscle, and really go into depth into what's changes. And so the database we have put in place allows you to really look at these changes at the molecular level. What I'll talk about today is really the GeneLab project started as a simple repository, but it has evolved so much from that point. We now are providing not only raw data, which would be very difficult for anyone to interpret and would require some computer power. So what NASA has put forward is actually a method to really turn this data into something meaningful. We have an advantage over other agencies like NIH is that we can really do it very precisely because our database is small, because you don't have that many samples being flown on the space station. It's just a very expensive enterprise. And for the same token, that's the reason why we want to look at everything that's being flown, not only humans. So the database actually is mostly focused on animals, plant, microbes. So we don't really have any astronaut data on the database. This is a separate entity that is dealt by JSC. So what we focused on is really we're very keen on trying to see what are the changes that are happening in a universal manner. If you look at, you can think of the space problem at many layers. We're all made of cells, and we're all made of DNA, so you can think of like fundamental changes at that level. And that gives you a glimpse of the system. But the problem is a biological entity like us is a very complex machine, and it's a very complex system. And so you need to understand how perturbation in one cell now affect the entire system. I'm sorry, so the video is playing. There was actually a soundtrack with it, but so I didn't realize it was playing. You want to restart it? Thank you. So just also to emphasize some aspects of the work that was discovered. So you've seen some of our students. We had an internship last year. So we put in place something called Analysis Working Group. So those are groups of volunteers from the scientific community. They're pretty much across the US, but also 4 different countries at the moment. And so we work together. We meet monthly in different groups. There's a group focused on plant, a group focused on microbes, one on all the animals, and then finally one on bioinformatics, state-of-the-art technology. And so together we look at the data and we try to interpret the meaning of the data. So the goal here is that the whole is bigger than the sum of the parts. So really it's by putting those data together we'll be able to pick up universal changes. biology is still a field that is growing, and I'm a physicist and did a lot of math, and so that's why I'm doing omics now. But the biology is just at the point of really reaching what we've done with physics. Numbers are starting to come a powerful tool for biology, and I think the omics is one of a reflection of that. And so really the goal here is to really standardize the process as much as we can and try to understand what's happening. So for instance, what we discovered that was not known is that we see some changes at the mitochondrial level in the animals that seems to be having profound effect in the liver. And this liver itself then sends signals into the entire tissue in the mice and seems to affect the eyes, the muscle, and so we're not really sure yet what it means, but it was a surprise because liver has never been really on the, if you look at the HRP, the Human Research Program, we focus on cancer, heart, and CNS, central nervous system. We never look at liver. And liver was just a tissue that was in the database that we just started looking at, and we keep seeing those weird signals. And sure enough, we talked to the scientists working with us, and they say, oh, we see in the eyes the liver signatures, and suddenly we start seeing this liver everywhere. And it was a bit of a surprise. We're still trying to tease out what's happening, so that's why I don't want to talk too much about it, because there was not a peer-reviewed publication yet on that topic. But this is the kind of things we're really excited about. And I think I should leave it there and maybe answer questions.
Great. Okay. So we'll pass a mic around. Kindly state your name and affiliation before asking a question. And thanks to all of our speakers for your nice remarks. So we'll start up here. If we could bring a mic up front. Thank you. And we have viewers watching remotely as well.
Thank you. Thank you. Seth Borenstein, Associated Press. For Craig, in terms of especially the twins experiment, looking at some of the results that are coming out now with the immune system, the mitochondrial changes, does this encourage you, discourage you for the long duration flights that you're going to be seeing to Mars? Is there now a new thing you're more concerned about as a planner that you weren't there? And is there something that you are less concerned about now?
Thanks for the question. As Chris said, this is a study with only 2 individuals, and so we don't regard any of this as conclusive. But on the whole, it's encouraging because, as Chris mentioned, we see a number of changes, most of which revert back quickly upon return to Earth. There are no major new warning signs. We are seeing changes, though, that we didn't necessarily anticipate. So are those consequential or not? We don't know yet. We will need follow-up work. But on the whole, I would say we're quite encouraged. And I'll also note that our Russian colleagues have flown for a year or more. 4 of them have. And so we know it's possible, but we also aim for more than just possible. We don't want the astronauts to just survive. We want them to prosper and to be able to perform very well. So we're also interested in something that may not be a health decrement, but is maybe impinging on performance. So one part of the twin study that the panel here today is really emphasizing the molecular, but the twin study also looked at some physiology, like fluid shifts in the body, and cognition. So to our knowledge, the twin study is unprecedented in looking at those different levels of biology. And happily, we didn't really see any changes in Scott Kelly in flight in terms of cognition. Interestingly, after flight, there's his very busy schedule and some changes that might be due to that. But basically, the short answer is there were no warning flags raised. There are some changes that weren't anticipated. We don't know if they're consequential or not, and we need to follow up now with more studies looking at those things to see if it's really widespread and if there's any impact.
Question here in the front. Yeah.
Thank you. I'm Alan Boyle with GeekWire, and I just wanted to follow up on that. I guess I'd have to review the research, but just for the Cliff's Notes version, could you talk about the changes that were unexpected and the changes that have persisted? I think you mentioned, Christopher, that there were some changes that have continued to persist longer than the others.
Yeah. So I think a lot of the changes, particularly observed, in flight, were related to things that you might expect. So things like DNA repair, we saw a lot of actually work on changes in mitochondrial respiration, which I also briefly described, indicating sort of, muscles responding. But the immune system was one of the sort of class of genes that seemed to be the most highly adaptive, and so changing the most. And also the things that really seem to be persisting. I mean, some of this is coming in subsequent papers, but we've seen that it's similar networks of genes. Like the immune system has a bit of a I don't know if it's definitely in the categories of, say, a immune system and DNA repair, the things that we see that have been, really activated and seem to be have some memory of what has occurred. So, and I think, as actually Dr. Kundra was just saying, is that there's probably no red flags. I think it's maybe just, mostly green flags and maybe a handful of things that are, I'd say, like yellow flags, things just to keep an eye on. But nothing that we haven't seen in other, to give it context, which is the most important thing for any of the changes you see in anybody, is that we see some of these changes also for people just on Earth. And so it's not as if we saw an entire subcategory of genes that have never been observed in a human being before. So I think that's why we think they're interesting, because we see that they're adaptive and dynamic, but things that are to be some degree expected. And one thing that was also discussed at the HRP meeting or last year was actually that there was a vaccination that was done in space, and all indications seem to be that the immune system is functioning fine. So we don't think as if it isn't even necessarily indicative of dysregulation. I just think it's more hyperactivation would be how I would describe it, or a high alert, I'd say, not necessarily dysfunction.
Is that the yellow flag?
Yeah, or yellow flag, just because it clearly is. It's one of the questions I often get is, well, are these things, if genes are going up, should we then give very targeted gene therapies to turn them all back down? And the answer is probably not necessarily, because these might be how the body actually adapts to microgravity or zero gravity. There's an old joke that microgravity is a bad name because we can't measure gravity that well, but less gravity, as it comes up at some of the NASA meetings. But it really is, I think, just something to keep an eye on, I think, for the future missions.
Yeah, two things. I think, Chris, you've previously described the changes to be in the same ballpark, roughly speaking, as like when someone undergoes scuba diving or mountain climbing type thing. So it's in the realm of, they're not like they're not pegging the needle. They're within the realm of, scale of changes that we see elsewhere. And the yellow doesn't necessarily mean bad. It just means it's change we didn't expect and don't understand. And it could be a good adaptive response to spaceflight with no permanent consequences, because you expect the body to make some adjustments. Or it could be setting things down a path that would be a concern, and we just don't know yet.
Yeah, and the other thing on like the mitochondrial levels in the blood was one of the ones that we just didn't expect. Something we know can change and again fluctuates in normal individuals, but seeing it spike in the beginning part of the mission was something that was unexpected. Yeah.
Question back there.
Thanks. Maggie Fox, I'm just following up on what you've just said. Can you talk about some of the DNA repair genes that you saw a change in function? And you're all talking about microgravity changing. Could there be other factors? I'm thinking cosmic rays as well, right? What else might be affecting this gene activity?
I'll take the latter part and then turn it over to Chris for the DNA. You're absolutely right. There are a host of things going on in spaceflight. So the most obvious, especially when you take your tourist trip up there, is the weightlessness. But you have the radiation. You have an altered atmospheric environment. There's higher CO2 levels on board. You have maybe a little stress associated, with the events. You are in from a cognition perspective, you're in this very limited environment, the size of a house, for 6 months, or in Scott's case, almost a year.
Yeah, there's no Netflix, right? That's up there.
Well, they do actually have access to video and stuff, but I don't think they've got the full. We're not paying for the. Got to be careful with making jokes. So you're absolutely right. There are a multitude of things, and their work schedule, and doing EVA spacewalks, et cetera. So that's another aspect of this work, is when we do see the changes, is trying to trace them back to the causes. But I'll let Chris elaborate on the DNA repair aspects.
Yeah, so the pathways involve the most mechanisms for DNA repair. So, site-specific DNA damage, homologous recombination, the normal DNA repair pathways that you would observe when DNA is damaged, say, from ionizing radiation. And so, but for the specific genes, they haven't been approved to say exactly which genes are upregulated, or what is the specific DNA damage repair gene. So I can't yet comment on those till the paper comes out. But we do have, when this paper finally comes out, which should be very soon, list of, each pathway, each gene, which ones are the most upregulated, which ones are going up as well as down. So those will be released with fine detail very soon. Yeah.
I want to add to this. So that's really where, a database like GeneLab becomes useful, because in our case, what we have is we have all these animals data. So we have ground experiment that goes along with the space experiment. So we have experiment on the database that shows you animal exposed only to cosmic radiation. And then we have also ground control. So for instance, one of the stressor is carbon dioxide. So what we looked at is when we looked at the liver in the animals, and other tissue, like to muscles and so on, we see a clear, strong carbon dioxide hypoxic signatures in those tissues at the molecular level, with gene expression being changed and linked to this. So the way it was done, I will talk about it today, is we have ground control where the animals are put into what they call an environmental chamber. It basically replicates all the telemetry on the space station, so temperature, humidity, life cycle, carbon dioxide. And so when they do these controls, you can go to 600 to 3,000 ppm. So here on Earth, we now are at 380. We used to be at 300. And so basically, 600 starts to be pretty high. But physiologically, there's no known impact of this level. But molecularly, we really peaked in a non-biased manner hypoxic signature in the tissue, which then begged the question to what does it do? We know on the short term, we see nothing in terms of phenotype, but what happens if you stay longer? And then the next question is, what happens if you put CO2 and radiation? And so this is the kind of thing you can do on the ground. So the ground experiments are critical to really tease out or deconvolve the signals that you're getting from these experiments or those data from the space station.
But one thing that's exciting is we've been starting to work together with data from the twin study in GeneLab and compares that there's brewing ideas that there might be a sort of conserved mammalian response to spaceflight that is not. We're in the middle of working on that paper now, but we do see some common signatures between mice and human models, and then also the mice data give us a great way to separate out what is just radiation. So it's coming along, I think.
Question here in the second row, Alexandria.
Thanks very much. Ivan Semenek with the Globe and Mail. I was hoping you could expand a little bit on the radiation situation, or just clarify whether the degree of difference between the surface and the ISS offers any information about cislunar space, or if it's just qualitatively too different, and also whether any of the biology experiments that are going to be announced for cislunar space speak to the radiation question.
Yes, absolutely. Thank you. So on the ground, in our day-to-day life, including visits to the doctor and the dentist, really the only ionizing radiation we're exposed to is X-rays and gamma rays. So they're high-energy light, basically. The International Space Station is above the atmosphere, which is one of our primary shields against against this particular, as in particles, radiation, so galactic cosmic rays in particular. But if you're on space station, you're still within the magnetic field, and you're not that high up. When you look at the geometry carefully, you have the Earth, the whole planet, shielding almost half of the radiation. So the radiation dose that the astronauts receive on the space station is less than deep space when you get beyond the magnetic field. And that's why these experiments on EM1 are particularly valuable, because we'll have over 3 weeks in the deep space environment and can see what kind of biological damage is being produced. So the dose rate in deep space is roughly twice what it is on the station. And when you get, and it's slightly different because the magnetic field is deflecting the low energy radiation, but not the high when you're on the International Space Station. And you're going to get it all in deep space. From ground, from normal ground exposures, you can't infer very much. NASA does have, however, at the Brookhaven National Laboratory on Long Island, we have the NASA Space Radiation Laboratory, where we can mimic these heavy particles. And we can't do the entire spectrum all at once, but we can come pretty close. But that's still, so it's a very valuable facility, but as always, there's nothing like the real thing. And so we're really keen to do these deep space experiments.
What are the questions for those deep space experiments? What are they trying to get at?
A common theme is the DNA damage to the systems being studied. But it's also, this data will be going into GeneLab, and there will be the opportunity to look more systematically. A great power of the GeneLab capability is you can, as a scientist, you go in looking for certain phenomena, but you're, with things like transcriptomics and other things, you're casting the net very broadly. So things may be changing that you don't anticipate, and they'll pop up. And so that's a great melding of the classic hypothesis-driven research along with the very broad capability that GeneLab provides.
Question right here.
Steve Marin, freelance for Sylvain or Christopher. Are there any of these changes, whether DNA or other, that you observed in the space environment? Do you have time resolution during the flight so you know how rapidly some DNA or other effect occurs?
Yeah, I'll speak to the human side, at least for the twins data, and there it's actually, pretty immediate, or at least as soon as we got our first sample. So we did have a lot of, just ideals that we'd want to get, a blood sample within the first 5 minutes of getting on the ISS, but that was not allowed. It was too complicated. They're in the middle of doing things once they get up to space. So, but within, to give you a sense of time, at least some of the first samples, within 48 hours of return to Earth, we saw a lot of changes immediately adapt. And then within, I think the first sample we actually got for the twin study was at the Launch Plus 7. I can double check, but like within a week. And so we don't know if it's within, a matter of minutes or hours, but we know certainly within days, responses changes on the ISS and also when he got back to Earth. Actually, especially the first 48 hours being back on Earth was some of the most dramatic changes we've seen. So, as soon immediately upon landing, we actually did one blood collection right before he stepped into the Soyuz, and then as soon as that was one where we actually were to get a blood sample as soon as he got back on Earth, and so we can see changes they're very dramatic and quick.
So for the animals, it's actually, the humans are better for this because body fluid for animals is very difficult. So to do a timeline, you need to do a non-invasive method, right? Or it would be, we only can fly about 40 animals, 40 mice at a time. So we only have very sporadic points. I think the future, to me, one thing that really excites me right now is that, ISS cases, for instance, is pushing very hard, and NASA also is pushing very hard for tissue on a chip. So those would be, pretty elaborate chip where you have different tissue talking to each other through little channels. So it's some kind of microfluidics. And I think this was going to be really interesting to look at because it's going to be like a mini human on a chip, and we can do all the omics on those. And I think we're going to have great time resolution. But right now we kind of limited for animals.
Question here, and then we'll come up here.
Hi, my name is Vijay Shankar. I'm a freelancer. My question is related to the effect on the biological clock. So I wondered if you have investigated on that side of, especially in humans, because there's jet lag, sleep cycle changes, and there's also other environmental effects or impact in the space. So I wondered if you could, enlighten us on that.
So the circadian rhythm is absolutely one of the things that we are concerned about in spaceflight, because when you're on the space station, you've lost the normal day-night cycle cues. And so actually, because of that precisely, we have changed out the lighting system on ISS to tunable LEDs, where we can change the wavelength. Because the strongest entrainer of your circadian rhythm is light. In addition to the rods and cones in your eyes, you have other receptors that are detecting mostly blue light, and they're entraining your circadian rhythm. And so we are using that to advantage, so the the astronauts can remain in a good circadian rhythm. We've done ground studies in collaboration with the Russians where in the absence of those cues, some people, their circadian rhythm remains just fine. Others very quickly went into what was called free running. They lost lock, and then for a deep space mission, that's not just like, interesting. It means they're not functioning as a high level, and then socially, they're like, when they're out of phase with their crew members, that starts to deteriorate the team dynamics, et cetera. So that's an area that we do pay close attention to, and on station have learned to basically change the light system and provide a stronger entrainer of the circadian rhythm.
We have a question up here from Tina, and then we'll have one in the back.
Hi, Tina Say with Science News Magazine. I was wondering if you could talk a little bit about Mark Kelly's changes. Did he have any instances where his gene expression or other things changed? And if you were to do this in the future, obviously you're probably not going to have a whole lot of twin astronauts. Could you do the same sort of thing just within a person before they leave and then, during spaceflight and after they come back?
Yeah, good question. So Mark, he served as the ground control, and we also compared it to other publicly available data sets of gene expression data, for example, to know what is just normal changes. And then we did have another part of the study, a donor that was we would even just take blood and just purposely degrade it in some cases, because some of the blood draws we got from space were called ambient blood draws, which is then confounded because we had to get the sample, return it back to Earth in Kazakhstan, repatriate the sample, which is a verb I've never used until this study. We had to get it back to Houston and then process the samples, and they're usually Fran, who was in my lab and now has her own lab, would be the one separating out cell types. And so, for example, our concern there is the gene expression might be also impacted by just transit, by just moving cells from space back to Earth again. So we did both frozen cell types that were sorted and also fresh blood draws to try and account for that, and using the same centrifuge conditions on the space station on Earth. But all that is just background to say we tried to normalize things as best we could, use controls everywhere we could, and look at all the changes and use Mark as a control as well as some other another donor. And he did, of course, have changes in gene expression. All of you have changes in gene expression, every day. It's really one of the main mechanisms how the body adapts and controls epigenetics to then control gene expression to adapt to really anything whenever you encounter any stressor. And we did, but so then we normalized all of the changes we observed in Mark and subtracted those from what we observed in Scott in a longitudinal fashion, what's called a generalized linear model, and then compared those two. So, but we did see changes in him that you would just expect for someone just living a normal life. So, for example, his immune system also, we saw changes where some genes would go up and come down, and so just not as much as what we saw in Scott. We did also try and get Mark we asked in the beginning of the study, well, maybe Mark will have the exact same food as Scott in space, because that would be a great control. He politely declined, and I think stuck to the nachos and margaritas and kept a food log. But, so that was one thing we were hoping for in the design of the study, and he just said, no, thank you. So, but we controlled everything we possibly could. And one of the things that came out that, there's two other groups I'd mentioned this. So Mike Snyder, as one of the other twins PIs, he has ongoing studies looking at many, many people longitudinally, so that healthy people. So we have a lot of data in the scientific literature about people with cancer, infection, or acute disease, but very, very little data about just someone that's relatively normal longitudinal. And so we have some data from what's called an IPOP cohort from Mike Snyder's lab, and also Andy Feinberg's one of the other investigators. And their epigenetically, for example, work from his group showed that it seems like Mark and Scott were relatively similar over the course of the study. And so that they seem to be, if anything, Mark, seem to be, they're about the same. So it's not like there was dramatic epigenetic changes in spaceflight. So.
Yeah, so in the classic scientific study, you're trying to control for as many variables as possible. And like Chris said, Mark was not willing to eat space food for a year, sleep when, Scott's sleeping, et cetera. And so if you look at it a different way, it's actually another kind of experiment where with Mark's free-range lifestyle, you get a good sense of what the normal variation is. And so when you see a difference between Scott and Mark, you have a good calibration for, is this like a significant magnitude, or is it just in the realm of normal fluctuations? So in a way, that's a good way to make lemonade out of the inability of the lemons of not being able to do the tightly controlled experiment.
A question in the back. Yeah.
Thank you so much, Joel Achenbach, Washington Post. Can you, a couple things. You said there's an embargoed paper coming out. Is that this afternoon?
Not this afternoon, but very soon. I don't have a
Okay.
final date, but it should be very, very soon.
Okay, and there is this announcement this afternoon about the experiments on EM1.
Correct. Yes.
Okay, that's embargoed until when?
Sometime this afternoon.
Okay, thank you.
Stephanie?
Okay, so
I can send you the link when it comes.
fantastic.
If anybody wants to come find me.
That's great. So, Chris, can you explain these dramatic changes you saw within 48 hours? I think you sort of answered it already. It's changes in gene expression. What does that mean? I'm sorry that I don't really understand what it is you're talking about. So can you, as simply as possible, explain what it is that you saw?
I mean, I'll comment on. So that, yeah, here there's lots of things in the paper and in the supplemental figures, and we measured, everything we could. So this includes every metabolite, every small molecule, every gene that we could quantify, and there the changes really are primarily the immune system itself. When I say the immune system is changing dramatically upon return to Earth, and also a lot of other small molecules that I can't describe yet until the embargo lifts, not today, but in a few weeks. Is deal a lot with how the body accounts for sensing what appears to be an immune stress, and then also inflammation. So we see all those signatures rapidly change, but it's really an adaptation. So suddenly the body is back on Earth, and even just the stress on the muscles is what we're seeing. That just the physical, the sheer stress of being back on gravity is something we see in the gene expression data as well. But I mean, when you say, I don't know if that quite answers your question. Is it?
So you see it in the blood. You're looking at the blood sample, correct?
Right.
Are you also doing other kinds of diagnostic measurements?
Yeah, so it includes everything. So the things I'll discuss briefly today is most of the work just on expression data, some on genetic information, and what we see in cell-free DNA. So that was what I mentioned. The mitochondrial DNA, for example, that we observed spiking actually in the beginning and at the very end of the mission, is something. So all of you right now are shedding DNA into your bloodstream. So as cells die naturally, they'll die, and DNA and RNA gets lysed in your bloodstream. And so we took what is present for the catalog of DNA and RNA in the blood, and then measured what was there. And so that's something we could observe. The genetics there of cell-free DNA, or just DNA floating in your blood, is an indicator of overall health. And so, and also just of which cells are dying at which speed. So. What we observed is that the mitochondria means that there's lots of, what we saw in the early flight, it means that there's a contribution either ejection of mitochondria or other cells that have lysed and released their mitochondria. And in the study, we can't discriminate them, so we don't know, but we just know that there's more mitochondria, for example, that we see in the bloodstream. And that actually is something I'll talk about this afternoon, that itself primes the immune system. So if your immune system suddenly sees a lot of mitochondria, it thinks that it actually views mitochondria as a different, because its DNA looks different than human DNA. So the body adapts to it as if it is kind of like a different genome in the body, which technically, evolutionarily, it kind of is.
And Chris, I might be missing something here, but another way to look at it is in terms of the samples we collected. We got cells from, I think, 3 places. Blood, so we got mostly white blood cells. We got some from the cheek, and then I'm going to count feces as well. Yeah, stool. And that's mostly looking at the microbial species present in the gut. And then there were more like global samples looking at the blood plasma and what molecules are present there, like Chris was just discussing, and urine. And both of those, in a way, are interrogating the whole body, giving you a picture of what's happening across the whole body. So in some cases, we were able to look at specific cells and what's happening with them. Other cases get an indication of what's happening across the whole body.
Just a technical point, just to clarify about gene expression, because I think that's always a confusing thing. The way I explain that typically is every cell in your body has the exact same DNA, identical. So one wonders why we have hair, eyes, nose, what's the difference? The difference is in the gene expression. we have a way to turn on genes in a way that will really reflect the functions. So when we see a small change in gene expression, it tells you that the cell has switched its status to a different status, and typically they are associated to specific activities. So it is a new way to look at things with respect to what we used to do in the past, where you do physiological measurement to the doctor, you look at the final endpoint. Those gene expression are kind of like precursor to all these things. And so they're very powerful, but they also have to be taken with a grain of salt, because you have to think of it in the context of what you're looking at.
All right. If that's all our questions, thank you so much to our speakers. That was excellent information. We appreciate it. Thank you.
Thank you.